that very kind of introduction. Yeah, I'm here. I should start by introducing my colleague, Dr. Naveen Nambudri, who I'm here with. Naveen's a real marine biologist. I'm often introduced as a marine biologist, and I say I'm a fake marine biologist because I work with marine turtles, and I work on them when they come ashore to nest. So marine things, but from land. But working with Naveen over the last few years, I've managed to get into the water a little bit, and Naveen and I work together on marine biology at hello, another marine turtle biologist. So. On marine biology at the IISc and on the interface between biology, conservation, and management at the Dakshin Foundation. Today, I'm not going to be talking about either marine biology and conservation. A lot of the work that I do at IISc is centered around ecology and evolutionary biogeography. And over the last few years, we've been doing this project trying to understand the evolution and diversification of herpetofauna in the Western Ghats, which is this mountain chain along the western coast of peninsular India. So I wasn't really sure, what the background of the audience was going to be. So I'm just going to run through a very quick theoretical background to some of the work that we've been doing, and much of it is framed in the framework of macroecology, which is a discipline that the term was coined about 25 years ago by James Brown, but it's a discipline that's become increasingly attractive within ecology. And macroecologists will claim that macroecology is important because it answers central questions related to abundance, distribution, and diversity of species, such as diversity along gradients, body size, range size distributions, commonness, and rarity. But more importantly, can connect different levels of organizations, both from individuals to populations to biotas, but also at different spatial and temporal scales. It's also been argued that understanding these patterns is central to conservation relating to conservation, but I think that's an entirely different debate by itself, so I'm not going to get into that. People have asked these questions, of course, for the last 200 years, from the time that even prior to Linnaeus's interest in the ecological relationships of species and the work during Darwin's era, questions such as, why are there more species in the tropics? Why are some taxa more diverse than others? Why are some rare? And why are some, for example, small body? These are basic questions that people have been asking for a long time. And macroecology has attempted to sort of bring these together in a large synthetic framework. Specifically, though, the patterns that we've been interested in are gradients along latitudinal and altitudinal axes. There's also been some amount of work on bathymetric gradients, but not so much, primarily because of sparser data sets. And the mechanisms that people have postulated over the years fall into 3 or 4 broad categories. The broadest one is environment-related drivers of richness, historical factors, evolutionary factors, but more recently, factors relating to the geographical range of species, which has become our central area of interest. In general, the patterns have been that lower latitudes have higher species richness, but most gradients, in fact, appear to show a mid-domain peak in richness. Most elevational areas, in most elevational gradients, peaks in species richness are in the middle elevations, and if one looks at the globe as a domain, again, the center of the domain has the peak in richness. The way that these have been typically studied are either in terms of the influence of a variety of factors such as climate topographies and species-related traits on species geographic ranges, or directly the influence of, say, environmental variables, area, and range-related factors on species richness itself. But the way that one way to look at it would be that all of these factors affect species geographic ranges, and species geographic ranges sum up to provide richness patterns in particular regions. Whether some of these variables influence richness itself directly or not is still an open question. There's half a century plus of work on species area relationships. There's half a century or more of work on the relationship between environmental factors relating from, again, in broad categories, solar energy, wind and rainfall, soil and productivity on richness and range. But About a decade and a half ago, ecologists began to look at the role of ranges themselves influencing richness patterns in ways other than might be mediated by the effect of environment on geographic range. And this came about through a couple of papers by Stevens in the 80s, where he came up with this hypothesis called Rappaport's rule, where he argued that latitudinal ranges increased with latitude. There was a positive correlation between latitude and range, and this he proposed was because temperate species inherently had to be adapted to a wider set of environmental conditions, and because of seasonal and annual variation in environmental conditions, they were naturally adapted to larger environmental ranges, and therefore were able to colonize larger geographical ranges, and the intrusion of these ranges into tropical areas, he argued, was increasing richness in tropical areas. This, in turn, sort of led to a hypothesis about how geographical range itself might influence species richness. In the 90s, Robert Caldwell came up with what he called the geometric constraints model, where he said that if you take a domain, a range, and throw in a bunch of random species randomly with randomized range sizes and range positions, you would and the range limits were constrained by these boundaries, you would end up with this hump-shaped pattern and richness without invoking any environmental factors. They also played with a few alternate models with empirical range size distributions and midpoints, but pretty much the patterns were that you got some kind of hump-shaped pattern with a peak and richness in the middle. And what they were basically arguing was that you could therefore explain a tropical species richness peak without invoking any environmental factors such as temperature, rainfall, topography, area, any of those things, that this would come about just by randomly distributing species across the globe. And I'm not sure if you've seen this, but Robert Caldwell had this little program where you could, let me set up bin size, where you could play with this model of his, and so basically this is a domain, and all that this program is going to do is add n species randomly. Let's throw in 25 species into this, and what it's doing is each row represents the range of a particular species, and it just throws in 25 species that have random range sizes and random range midpoints, and you can do this over and over again, and you will get patterns that are largely hump-shaped in their across the domain. So essentially, some ecologists were arguing that these patterns and species richness that you got in latitudinal and latitudinal gradients were an outcome of entirely neutral processes, or could be modeled as outcomes of entirely neutral processes. But of course, as biologists, we know that environment does make a difference. This is a controversy that goes back a while. There's been controversies ranging from issues in island biogeography to these mid-domain models about whether non-biological explanations can suffice, when we actually, as biologists, know that biology matters. What the proponents of the Null models were arguing was that even when you didn't use these neutral models, you were actually implicitly assuming a background model which was of uniform variation, and that uniform variation may in fact not necessarily be what's expected if there are no biological factors at play. Now, the problem with the initial approaches to analyzing these patterns was that people took richness as a response variable. They correlated with environment, area, but also with predictions of these mid-domain models. And this was really not a very rigorous approach, because the predictions of the mid-domain models were actually predictions. They weren't predictors. So this approach was criticized because in these sort of multiple regression frameworks, some of the predictors were some of the parameters were predictors and others were predictions. There's also a number of other caveats. And I'm going to just show data from some of the work that we did in the Himalayas using a plant data set. Patterns vary across scales. When we analyze patterns from local to regional level scales, we found that there are different explanatory variables at different scales. We found that patterns vary across functional groups. So when we divided these groups as temperate, tropical, and cosmopolitan plants, we found that there's a pointer on this one, right? Yeah. So we found that for tropical groups of plant, area was a really strong predictor, temperature was a really strong predictor. For temperate plants, on the other hand, temperature continued to be a strong predictor, but not area, not productivity. In fact, the predictions of the mid-domain models were most closely predicted with it. So patterns varied with groups that we often considered as single groups. So when we, even with other taxonomic groups, when we study frogs or reptiles, we often tend to cluster them together in these taxonomic groups, which might actually be showing different responses within them. And finally, we also proposed that pattern does not imply mechanism. I won't go into too much detail of the model, but basically what we did was we hypothesized, we created a model where at the level of a single hill slope, there were certain environmental mechanisms or neutral mechanisms that influence the distribution of tropical and temperate species, and we summed those patterns across scales. And what we found was that if you assume that both tropical and temperate species were correlated with, were determined by temperature, you could sum up across scales, and at the largest spatial scales, you would still get a hump-shaped pattern. You could hypothesize that that both of them were not influenced by any environmental factors, merely by neutral mechanisms, and you still got a hump-shaped pattern at the largest spatial scales. You could combine the effect of temperature on tropical species and assume that temperate species had no environmental effects acting upon them, and at the larger spatial scales, you still got a hump-shaped pattern. So regardless of what was actually driving species distribution patterns at local spatial scales, which is where you'd expect environment to act, at the larger spatial scales, when you summed patterns across the entire Himalaya, you still ended up with the same pattern. And just, a cute outcome was that the model that combined neutral mechanisms for temperate species and temperature for tropical species look not unlike the actual patterns that we got at the largest spatial scales in the Western Himalaya. I wouldn't put too much weight in this result, but it looks pretty similar. Now, moving on from there, what clearly was required were models that would combine the effect of these range overlaps that was becoming increasingly obvious that range could play a role in determining species richness patterns. In 2007, Kartik et al. Came up with a set of models which would combine the distribution of, which would model the distribution of organisms based on the assumption that ranges are contiguous, which is essentially what these range models were postulating, that if I take a bunch of contiguous ranges and throw them into these domains, they would create these hump-shaped patterns. So we modeled the distribution of organisms as if ranges are contiguous and as if they are not. And these they called range scatter and range cohesion models, and combined the scatter, which is just discontinuous ranges, and cohesion, which are contiguous ranges, with environment. So in range cohesion models, for example, you start out with a random location for a species, and in the next your next modeled location is contiguous, so it's one of the adjacent grid cells, but correlated with an environmental variable of your choice. So when this could be rainfall, it could be temperature, it could be any factors. So you could come up with a distribution that was simultaneously contiguous and incorporated rainfall and temperature. You could do the same thing, correlating it with rainfall and or temperature, but without any contiguity. And in essence, what people had done for the last 100 years was a comparison with what had been created as a range scatter model. So you assume that species distributions of species richness will be correlated with temperature regardless of where they are in this global or regional domain that you're studying. And I discovered, to my amusement, that there's actually a law that defines range scatter, and it's called Beijerin's law, and it says everything is everywhere. Robic et al. In 2007 built these models for South American birds, and they used a combination of range cohesion and range scatter models combined with rainfall, temperature, and a bunch of combination variables, and found that some of the range cohesion models describe the empirical species richness patterns best. What this allowed one to do was to incorporate this element of range, and if in fact the range scatter models work best, then what one would have demonstrated is that in fact everything is possibly everywhere, and if the range cohesion models work better, then you infer that these range overlaps actually play a significant role in describing species richness patterns. So with that background, this is largely the framework that we wanted to apply to our understanding of the distribution of herpetofauna in the Western Ghats, both to understand their origins and their distribution, and also to understand richness patterns across the range. And what I'm going to present in this part of the talk is the beginning of that work. We've had to start with the delimitation of herpetofaunal lineages, because herpetofauna in general in the region and throughout Asia are fairly poorly understood in terms of well, before I go into that, just a very brief description of this group. Frogs, snakes, and lizards show the highest percentage of endemism amongst vertebrates in the Western Ghats and many other parts of the world. 80% of the known frogs in the Western Ghats are endemic, 60% of endemic lizards and snakes, and there's 100% endemism in many genera, so very interesting group for us to look at. As I was saying, while wanting to address these broader questions in richness and diversity, we were faced with two problems. One is the Wallacean shortfall, which is the lack of data on geographical distribution at various scales, and this is widespread across smaller vertebrates, small mammals, nocturnal birds, invertebrates, and so on. And apart from our academic interest in this, the data is obviously crucial for things like for conservation assessments, reserve design, and other applications. A second problem is the Linnean shortfall, which refers to the presence of a large number of new species, mostly in the tropics, and of course in marine ecosystems, and the poor knowledge of the taxonomy of these species. And these two problems had to be solved before we could address these larger questions. Now, what's interesting about what was interesting to us, or what challenged us, was the ability to address this, was the role of a Wallacean approach in addressing this Linnean shortfall. Now, typically, Linnean shortfalls have been addressed using morphological taxonomy over the last 200 years, but more recently, molecular taxonomy, and in certain instances where possible, other traits such as skulls. And of course, this is because there's been a long and unresolved debate about what species are, from the time of Darwin himself, who said he was struck by how entirely vague and arbitrary the whole distinction between species and varieties were. Dobzhansky, who pioneered the idea of the biological species concept, himself, ranted about the futility of finding this universally valid criterion, and as a consequence of that, a list, a shopping list of species concepts, some of which I actually heard for the first time when I saw this table. But we decided to use what Kevin Kuros created as a generalized lineage concept, where he argued that diverging lineages go through different stages in terms of their molecular, morphological, and trait divergence, and often different species concepts are addressing different stages of divergence. So we were just looking at lineage divergence in our research. Now, what people typically do in discovering and delimiting diversity is they go out and do surveys across a set of locations of interest. There's a bunch of described species. They pick up things that look different and compare those that are different with those that are known, and describe a few new species. We decided to abandon that approach, and what we did was instead to capture in our sampling the entire range of topographic, bioclimatic, and geological heterogeneity. We classified these massives, which range from 0 to 2,600 meters, across 4 different mountain complexes and sampled almost all of them. We still have one left to go. We also sampled across all elevational gradients in these blocks, and across the elevational gradients, we sampled a range of climatically different habitat types. So in a sense, we placed a grid over the entire Western Ghats and went out and sampled every topographically, elevationally, and climatically different region. What we got was, we had a total of 250 odd sites, over 2,000 data points, and lots of samples. What we then did was, we took a multi-criterion approach to lineage delimitation, and in general, species recognition has been based on a single criterion, and more recently, as I said, the advent of molecular tools has allowed us to incorporate phylogenetic frameworks. What people usually do is compare a new morphological type using molecular data with something that they think is its closest relative. What we did instead was we started with the haplotype phylogeny, and in our phylogeny we had, to the best of our knowledge, most of the lineages that are still extant in that geographical region. So we didn't know who was related to whom. We didn't know what our basis of comparison was to begin with. We just had everybody in the family tree on our tree. And that allowed us to at the second, in step 2, to make those genetic comparisons with the closest relative of each individual lineage. We used then morphological divergences-based cutoffs for lineage selection, which is standard, but we also use geographical range overlap as a criterion. Yes. So, I have a slide with steps. So we identified these exclusive lineage, these haplotype clusters. We identified the sister lineages. We looked at the genetic distance between these. We used a cutoff, and the cutoff was based on 2 sympatric species that, from calls and from morphology, we knew were different species, and they were sympatric, and they had a level of genetic divergence which was 1%, and so we took 1% as our minimum cutoff. In 2 out of the 3 genes that we examined, lineages that met the divergence cutoff in 2 genes were selected for further analysis. Now, what one would expect is that lineages that people had, all of these herpetologists have been going around sampling for the last 20 years, that they would have picked up these lineages that were really separated from each other, and maybe they were missing these ones that were closely related. As it turns out, that is not the case. We found known and unknown lineages across the entire spectrum of divergence between sister pairs. So there were some sisters that were undescribed that were as expected, low divergence, and not very different from their sisters. But there were others that were highly divergent that still had not been picked up. We got a 100% increase in divergence, and of these 29 lineages that were diverged from their sisters by the minimum cutoff, 16 had moderate to high levels. We had every combination of these in this set. We had exclusive clusters which were highly divergent from each other. We had low divergence lineages that were separated in geographical space, that were allopatric, so they were completely separated in allopatric space. We had low divergence lineages that were sympatric, that occupied the same location, and parapatric that occupied locations along spaces along a gradient. Most of the lineages are allopatric, as you would expect in species with low dispersal abilities. We explored low divergent lineages, which is this set across here in geographical space, most being allopatric. We also explored them in morphological space and found that some of these low divergence lineages are completely separate from each other, and other low divergence lineages completely overlap with each other. We found allopatric and morphologically divergent lineages, genetically divergent sympatric and morphologically divergent lineages. So we got pretty much every combination that one would expect. And if you look at this as a table, you find that you have, if you look at them in space, that is allopatry, sympatry, and parapatry, morphological overlap and lack thereof, genetic distance to various degrees, you have species that fall into almost every one of these categories. So there's no direct relationship between geographical overlap or lack thereof, morphological divergence or lack thereof, or genetic distance between these lineages. We looked at this across a bunch of other taxon. I don't have such detailed information for these yet. We're still analyzing them. But for Macroecology, which is another, the tree frogs that I just talked about are fairly recently evolved. They evolved about 20 million years ago. Macroecology is one of the genera that evolved about 100 million years ago in the Western Ghats. So Gondwan element. Again, similar patterns, new lineages across the entire range of divergence and across the entire phylogenetic tree. Indirana, another old group, large numbers of lineages, especially in the low divergence category. Nyctobatrachus, also 100 million year plus old genus. Wide range of categories of molecular divergence, and again, a spread of new lineages across all of them. Nemaspis, which is a gecko, actually a threefold increase in the number of lineages, about 16 described lineages at this point. There are probably about 50 plus lineages in the Western Ghats alone. Again, ranging from about 1 to 2% to 6% and above, which is really large. The random group that we looked at was particularly interesting because for all these other groups that I talked about, you could take a herpetologist to the Western Ghats, and a good self-respecting herpetologist would say, sometimes I'm not sure what species that is because this is a really divergent lineage. There's probably lots of new species. It might be that species or something related to it. For pretty much all the genera that I mentioned. With ranids, even a layperson would and I consider myself to be a reasonably layperson, would say, oh yeah, ranids, I know the ranids. That's temporalis, that's orange checker, and that's Curtipus. And yeah, they're widespread throughout the Western Ghats. They all look the same. I don't know that much about amphibians, but I can pick one up and identify them. So this was a group that we actually thought had very little variation in it. And exactly like with the other groups, there's absolutely no morphological variation. And yet, when you look at the molecular data, there's these different ranges of divergence. To the extent that the postdoc that I had working on this project, who's been working on this group for many years now, was actually picking up specimens in the field that he thought were temporalis and were turning out to be orangechecker, and the other way around. Which means that we're now getting sympatric species with low levels of divergence and no morphological variation. And this brings me to the point where I say, this methodology that we thought was great fails at that moment. So we've actually come up to a point with this that we know that there's divergence there, that even this method that we thought was really cool cannot actually discover it. We think it gets at pretty much everything else. What's an outcome of this approach to the research is will bring me back to what I talked about at the beginning. The kind of data that we've now generated now allows us to go back, plot these the ranges of these species in that geographical space. We know the identities of those species or lineages, and it allows us now to build those models that will allow us to understand species distribution and richness patterns. The point up to which I had brought that discussion was the range scatter and range cohesion models that were combining this 100-year-old endeavor to understand richness by combining environmental patterns, species area relationships, and range-based explorations. One thing that the ecologists had missed out was the role of evolutionary factors. And I've talked a little bit in the past about how one of the first lectures that I do for our graduate students is about how biogeography, ecology, and evolution actually all have the same origins. They start with the work of people like John Ray and Francis Willoughby in the late 1600s, and through Linnaeus and Cuvier and Lamarck and Darwin, biogeography, ecology, and evolutionary biology have basically one single 100, 150-year period of birth. Somewhere in the mid-1800s, they diverge. And biogeographers are looking more at contemporary ecological patterns. Evolutionary biologists are looking at microevolution. In about the 1960s, when MacArthur and Wilson proposed island biogeography, they had a chapter on evolution on islands, but they never incorporated it quantitatively into their framework. And over the last 2 or 3 decades, there's been discussion about bringing ecology and evolution together again. And so in this particular, in the macroecological framework, Gotelli et al. Proposed that we've talked about dispersal through these range models, we've talked about environmental gradients, but we haven't talked about evolutionary origins. And they proposed that we now needed to incorporate this third factor in. So looking back at that conceptual framework, the missing element in that was evolutionary history. But the kind of approach that we've taken allows us to do just that. It allows us now to take that information on species ranges, the environments in which they're found, the areas that they occupy, but we can now also infer where they originated. So in those models that I showed, we can where a species, which randomizes where a species originates, and then infers distribution based on environment and range contiguity. Now we have data that will allow us to input differences in where species originate and incorporate that into models, allowing us to incorporate all of these factors in looking at what drives richness patterns or diversity patterns in these broad geographical scales. Just as an aside, to demonstrate that there are more than just differences in origin, this group of tree frogs that my student Vijay Kumar studied, he found that there were these two very distinct clades in the northern and southern Western Ghats, which are separated by this gap. The gap is a few hundred meters in height, and the hills go up to about 2,500 meters. So it's a fairly deep gap. But over a 20 million year time period, you would actually be expecting a lot more exchange than there was. So it's actually been a fairly effective barrier for dispersal. But more than that, if you look at the biology of these species, most of the species found in the northern clade are open canopy species. There's been a lot of adaptation to grassland in those habitats. There are no subcanopy and canopy species in the northern clade. The southern clade, on the other hand, is a very close canopy clade. So the origins of these and the environment, the evolutionary and environmental history of these clades also reflects in their ecological and biological traits today. So in summary, we're moving towards these models that combine range, environment, and evolutionary history, which require detailed knowledge of distributions and history. And the frameworks, the sampling frameworks that we've developed over the last few years that address both Wallacean and Linnean shortfalls, incorporating geography as an explicit element, we feel provide the kind of information that's actually required to answer these questions. The hierarchical approach that we've taken to species delimitation or lineage delimitation is also relatively novel. With the examples that we've chosen with the model systems that we've chosen are already showing 200 to 300% increases in lineage diversity of lesser known taxa. And we're hoping that someday fairly soon this will lead us to this holy grail of understanding diversity and distribution at multiple scales. I'd like to thank all of my project team, which includes researchers, several PhD students, and postdocs, a bunch of other herpetologists and partners with whom we've worked, and a wide set of institutions as well as donors. In the last 3 years at least, a lot of the work that we did was funded by my institute and the Critical Ecosystem Partnership Fund program for the Western Ghats. And with that, I'll stop. Thank you.